Hello, my name is Natalie Arnson, and today I will be presenting my research project on the effects of stevia on insulin and the control of blood glucose levels. My research problem and theory discussed under my research problem is glucose and its adverse effects on the body, its effect on insulin levels, as well as a debate of artificial sweeteners. My research theory is regarding stevia's effect on blood sugar, stevia's effect on insulin levels, and is stevia considered a healthy alternative for a glucose substitute? I thought it would be helpful to first define what stevia is and how it relates to the concept of artificial sweeteners. Stevia, also known as Stevia rubetana bertoni, is a branched bushy shrub and belongs to the family Asaceae and is considered a native of the northeastern regions of Paraguay. It is also found in Brazil and Argentina. In addition, its cultivation is directly spread all over the world, including Canada and some Asian and European regions. It is known as a calorie-free biosweetener of high quality, as discovered in Jan A.'s study in 2021. In addition, artificial sweeteners can be defined as a sugar substitute that is a chemical in nature. It is added to some foods to make them taste sweet. It is made to taste like sugar or corn syrup. Usually, it has a much lower energy value than that of sugar. The third thing I want to relate to was the physiology of blood glucose fluctuation cycle and how it can be broken down into several categories. The first is that circulating glucose is derived from three major sources. First, intestinal absorption during the fed state. The second, glycogenolysis, which is the breakdown of glycogen. And the third, from gluconeogenesis, which we've discussed in class, states the formation of glucose is primarily from that of lactate and amino acids during the fasting state. So during the first 8 to 12 hours of fasting, glycogenolysis is the primary mechanism by which glucose is made available. Glucagon facilitates this process and thus promotes glucose appearance in the circulation. Over longer periods of fasting, glucose is produced by gluconeogenesis, which is then released by the liver. Glucoregulatory hormones include insulin, glucagon, amylin, GLP-1, glucose-dependent inolotropic peptide, also known as GIP, epinephrine, cortisol, and growth hormone. Of these, insulin and amylin are derived from the beta cells, glucagon from the alpha cells of the pancreas, and GLP-1 and GIP from the L cells of the intestine. In the fasting state, glucose leaves the circulation at a constant rate. Renal gluconeogenesis contributes substantially to the systematic glucose pool only during periods of extreme starvation. Although most tissues have the ability to hydrolyze glycogen, only the liver and the kidneys contain glucose 6-phosphatase, the enzyme necessary for the release of glucose into the circulation. In the bihormonal model of the glucose homeostasis, insulin is a key regulatory hormone of glucose disappearance, and glucagon is a major regulatory of glucose appearance. After reaching a post-meal break, blood glucose slowly decreases during the next several hours, eventually returning to a fasting level. In the immediate post-feeding state, glucose removal into the skeletal muscle and adipose tissue is driven mainly by insulin. At the same time, endogenous glucose production is suppressed by 1. The direct acts of insulin delivered via the portal vein on the liver, and 2. The paracrine effect or direct communication with the pancreas between the alpha and beta cells, which results in the glucagon suppression. I now want to discuss and dive deeper into the major research regarding my area of interest. The first is from Ajami M. and his other researchers, a study published in 2020 regarding the effects of stevia on glycemic and lipid profile of type 2 diabetic patients, a randomized control trial. So this trial broke down into three major categories. First, stevia and the glycemic index. Second, lipid profile. And third, the results regarding the enzymatic approaches of D-glucose calorimetric kits and blood cholesterol through Tinder CHOD/POD. So this major study discusses how one of the major things that was discovered is stevia's effect as a non-calorie biosweetener. The next major research was from Philippert K. and their other researchers in 2017 as steviol glycolides enhance pancreatic beta cell function and taste sensation by potentiation of TRPM5 channel activity. Stevioside increases action potential frequency in beta cells. This is first done when TRPM5 mediates a depolarizing current, which tunes calcium intake and oscillation frequency after glucose stimulation. Consistent with this, a glucose-induced cation current is potentiated by a stevioside in the pancreatic beta cells. Steviol and stevioside increase calcium activity in the islets. When stimulated with 10 milliliters of glucose, pancreatic islets respond with oscillations of calcium intake. In addition to this, application of stevioside resulted in a dose-dependent increase of the frequency of the glucose-induced calcium oscillations in the WT islets. Overall, the main effect is that in type 2 taste receptor cells, positive modulation of TRPM5 increases ATP release and afferent signaling from the receptor cell. In the pancreatic beta cell, among glucose application, beta cells display parallel VM and calcium oscillations which drive insulin secretion. TRPM5 is active during the lag phase in between bursts of action potentials and determines the frequency of VM and calcium oscillations, which modulates insulin secretion. Enhanced TRPM5 activity results in higher oscillation frequency, which results in more insulin secretion. The next area of major interest was done by Stamazaki, SN, and their research partners in 2020 regarding the effects of daily consumption of stevia on glucose homeostasis, body weight, and energy intake, a randomized open-label 12-week trial in healthy adults. So this experiment involved 28 healthy participants, aged anywhere between 25 with an increase or decrease of 5 years, perhaps, and they were randomized into either the stevia group, which was 14 members, required to consume stevia extract daily, or to the control group of 14 individuals. Glucose and insulin response at weeks 0 and 12, the glucose and insulin responses to an oral glucose tolerance test were measured. Body weight and energy intake were assessed at weeks 0, 6, and 12. There was no significance or difference in the glucose or insulin responses. The effect on body weight is that there was significantly a main effect in the body weight change, as the stevia group maintained their weight as opposed to the control group, which had a mean weight change at week 12. Therefore, then the energy intake results were analyzed. The energy intake was significantly decreased between weeks 0 and 12 in the stevia group. However, there was no change that was found in the control group. Another major research was from Jan A. and their research partners in 2021, the anti-diabetic activities of natural sweetener plant stevia, an updated review. This review discussed mainly the concerns with diabetes. So, their stevia jelly decreases the postprandial glucose level without any release of insulin, hence is recommended as a safe food product for the diabetic patient. The functional food is useful to provide nutrient requirements for the body and helps against other degenerative diseases related to today's changing lifestyles. The modified coconut jelly was prepared by replacing 50% of the sugar with that of stevia. This can also be put under the category of therapy for diabetes. Various extracts of stevia has been used for many years by South Americans as the therapy for diabetes. In diabetic patients, it also leads to hypoglycemia via lowering both the glycolysis and the gluconeogenesis processes, and by absorbing the glucose in the duodenum part. It has beneficial activities in pancreatic tissue by increasing the insulin level and enhances anti-diabetic properties in PPARY-dependent manner. And though its antioxidant activities, it works through. Moreover, the stevioside of stevia has been reported to lower the inflammation by lowering the level of pro-inflammatory cytokines. According to Jefferson, the stevioside increases the insulin level by affecting the beta cells of the pancreas and lowers the blood sugar levels. Another area of research was from Anton and their partners discussed in 2010 on the effects of stevia, aspartame, and sucrose on food intake, satiety, and postprandial glucose on insulin levels. The experiment contained 19 lean and 12 obese subjects, aged anywhere between 18 to 50. Food test days were done with a preload to stevia, aspartame, or glucose levels before lunch or a dinner meal. The preload order was balanced, and the food intake, kcal, was directly calculated. Hunger and satiety levels were reported before and after meals and every hour throughout the afternoon. Participants provided blood samples immediately before and 20 minutes after the lunch preload. Stevia preload significantly reduced those postprandial glucose levels compared to that of the sucrose preloads, with a P range of less than 0.01. Some of the issues. So, there were a few major issues presented within the study research regarding stevia's influence on blood sugar levels as well as the response to insulin. The first includes longitudinal studies. Further research is needed to determine if stevia's regular consumption brings sustained benefits for humans in a longer study. The next is a laboratory setting. One limitation of the present study is that the eating behavior was measured in a laboratory setting rather than the participants' natural environment. Similarly, the design of the present study was not in line with the typical eating patterns, which may limit the generability of our findings. The third was the control condition. Some studies did not appeal to control the data with a sweetener or a placebo. Human trials. Much research has been conducted regarding the effects of stevia, but many have not been done on humans, but rather on rats to understand its true effect. And there was an emphasis on the diabetic and obese population. Many studies lacked a standard normal population, which is both a benefit for implication, but a downfall for learning about non-conditional outcomes. Interpretation. So, as we can see, physiologically, everything can be supported by literature. However, genetic insufficiency, considering type 1 diabetes, normal populational outcome, processed foods, and connection to glucose and chronic health problems, as well as short-term versus longitudinal studies, should all be addressed. As we can see in this diagram, it is here to emphasize that from the data derived from the previous research, how and why stevia remains a healthier alternative than other known artificial sweeteners. Conclusions. Research would support 3 main emphases can be seen from the articles. The main research provided. 1. No effects on blood glucose, insulin, and lipid levels. Stevia is full of many important phytochemicals, steviol, steviosides, and ribosides that have properties to reduce blood sugar levels. Substituting sugar with low-calorie sweeteners like stevia may be a factious weight management strategy. Other research includes, although not placebo-controlled, these results suggest that daily stevia consumption does not affect glycemia in healthy individuals, but could aid in weight maintenance and the moderation of energy intake. 2. It possesses a high anti-hyperglycemic activity that serves as a substitute for saccharose in diabetic patients. It has beneficial activities in pancreatic tissue by increasing the insulin level and enhances anti-diabetic properties. It also helps in maintaining normal blood glucose and sugar levels by lowering inflammation and oxidative response. It is a major source of high-potency sweetener for the growing natural food market. The efficient function of stevia breeds, tea, jelly, etc., can provide optimum nutrients to the body, but also help maintain and controlling diabetes. And the consumption of sugar-sweetened beverages may be one of the dietary causes of metabolic disorders, such as obesity. Can stevia be a change? We're excited. Here's a list of my works cited from the research presented. I hope that you learned a lot about the effects of stevia and hope that you will consider using this product for future implications within your own diet. Thank you.